a reply in the chat room to let me know. Okay, so I can see one reply only. So how about the other student? Okay, so thanks for the reply. Okay, then I assume everything is working. So before we continue to talk about the body fluid analysis, okay, so let us have a brief summary for what we have been talking about somehow 2 weeks before. So 2 weeks before, we have finished the topics on the forensic toxicology. So we've ended up with the 2 miscellaneous cases. So the first miscellaneous cases, it is related to the carbon monoxide poisoning. So most likely it could be a suicide cases or an accident. Maybe it could be a murder cases. So in order to analyze carbon monoxide, normally we try to make use of the blood sample, okay? So because the carbon monoxide will bind to the hemoglobin tightly, so therefore, where you can find hemoglobin, most probably that will be in your blood. So for the respective analysis, so we have 2 different ways, okay, to conduct the carbon monoxide analysis. The first one that is by the use of the UV visible spectrophotometry. So with the use of the absorbance measurement, so we try to make use of the multiple analysis. Because in the blood, there would be hemoglobin binded with oxygen, and we also have the hemoglobin bind with the carbon monoxide, and we also have the hemoglobin itself. So therefore, we have 3 absorption species there. So with 3 unknown concentration. So that's why we have to make use of the multiple analysis by analyzing 3 different wavelengths. So we have 3 Bayes law, and then we have 3 unknown there, so that we can solve it simultaneously. So that is the use of the multiple analysis under the UV visible spectrophotometry to resolve, okay, the amount of carbon monoxide in blood. The other kinds of method that we can use, that is the GCMS. By use of the GCMS, most likely we try to couple with the head space extraction. So although we say that the carbon monoxide that is binded to hemoglobin tightly, it still could be volatile getting out from the blood sample if we can warm it up. So therefore, we can make use of the head space extraction to fill up with the blood sample, and we try to adjust the temperature to warm up the solution so that the hemoglobin will release out the carbon monoxide. So carbon monoxide will be volatile enough to migrate into the head space of the extraction valve. Then we just make use of a gas syringe in order to take up the gas and then inject to the GCMS or the respective analysis. So therefore, we have 2 different ways to deal with these kinds of miscellaneous cases. So carbon monoxide in blood samples, so by the use of the UV visible or the GC mask. So for the second kind of miscellaneous cases, that will be the drunk driving. For the drunk driving, we concern about the ethanol content. So by the use of the breath sample, okay, so that is somehow the sample that we need, okay, so for the ethanol analysis. So by the use of the breath sample, so probably I missed the R right here. So with the use of the breath sample, the equipment that we use could be classified into 2 types. The first one would be used for the on-site analysis. And then for the second type, that will be used in the lab-based analysis. For the on-site or on the lab-based, actually there are certain kinds of breathalyzer but the detector that we use would be quite different. For example, like for the on-site analysis, the measurement principle could be based on the calorimetry. So basically, we are making use of the potassium dichromate in order to react with the ethanol, okay, for a redox reaction. So induce with a color change, then we can measure by the change of color, or even it could be a fuel cell setting, because we have the redox reaction, so we have the electron transfer, okay, so we can measure by the current. So we measure ethanol, okay, by its property, okay, to participate in the redox reaction. So therefore, the result, okay, we can say that that is preliminary only. That's why, okay, that is an on-site analysis. So in the lab, we try to make it confirmative, okay, in order to confirm, okay, how much are there and then what it is, so that we can measure the ethanol directly by the use of the infrared spectrophotometry. So by the infrared spectrophotometry, we really measure the absorption wavelength of the ethanol so that we can quantify it directly, okay? So therefore, what you can be see, okay, for the on-site, we are not measuring ethanol directly. Somehow there will be an indirect analysis by measuring the respective redox reaction. And for the lab-based analysis, so that will be confirmative. So we have been measuring ethanol directly measuring itself. So that is the two miscellaneous case that we end up, okay, with the forensic toxicology. Then we start to talk about the body fluid analysis. And most likely the body fluid that we concern the most would be the blood. So mainly that is a very common kind of body fluid that we can find at the crime scene, okay, especially in some murder cases, or for some cases, some of the victims are being hurt, okay, so we can find the blood sample. So with the blood sample, we try to analyze by the respective blood group, okay, or we try to measure the DNA so that we can achieve the criminal identification. In other words, that would be the personal identification. So we can identify someone, okay, so by the use of the blood. So when we are seeing a red color liquid, or we can see some red color stain at the crime scene, so we have to answer the first question. So whether the red color liquid or the red color stain, okay, that is blood or not. So in order to deal with this kind of question, basically at the crime scene, we perform the scanning, okay. So we try to screen, okay, whether that is potentially a blood or not, okay. So with the measurement, okay, we say that hemoglobin will be a unique differentiable characteristic of blood. So with the hemoglobin, then we try to add in the first reagent of the hydrogen peroxide. So with the reaction, then we are resulted with the reactive oxygen. So with the reactive oxygen, then we further add in another reagent. So with the redox reaction taking place, then finally we can see a color change. So with a color change there, then we can obviously know that hemoglobin may be there. Then tentatively, it could be a blood sample. So the reagent that we use could be a Kastle-Meyer test that will be the phenolphthalein. So the positive result will be of a pink color, or we can be making use of the leukomalachite green. Then the color change will be of blue-green, or to be the most common one that we use at the crime scene, not only identifying whether the red color stain or the red color liquid that is blood or not. We also try to spray the luminol, okay, around the crime scene in order to check out some of the blood stain that we have been missed out, or some of the blood stain that has been cleaned, okay. So with the luminol, so we can see the blue fluorescence, okay. So these are the color that we can be result, okay, with the screening test. So most likely there will be certain kind of color test there. Then when we go back to the lab, then we try to perform the confirmative test, and then we try to add in some reagents, okay, towards the sample that we have. So in order to build up some crystal, so that we can observe the crystal under the bright field microscope. So most likely that will be the Takayama test or detachment test. So add in some reagent, then we can see some brown color crystal. So if you are using the Takayama test, the crystal, the shape that will be further reshaped in a cluster. If you are using attachment test, then that will be regular shape with a rhombic termination. So with the confirmed, okay, the red color liquid or the red color stain is blood. Then the second question then we need to ask, that is whether the blood that is belongs to human. So last time we end up with this kind of preliminary discussion. So what we know is, in order to check whether that is on a human origin, we try to make use of the P-Civitan test. So with the use of the P-Civitan test, what we are doing, that is in the blood, okay, if it is coming from human, then we can find some human antigen there. Then we try to add in an reagent. So that is the anti-serum, okay, that is of a known origin. So let's say that is from human. So if it's an anti-serum from human, if we can be finding the human antigen there, actually we will be seeing a precipitation. So that the precipitation shown, okay, that will be a positive result of the P-Civitan test. And then you can know about the origin, okay, by the respective anti-serum that you add, okay, to the sample. So last time, okay, we end up with this kinds of preliminary introduction. And for today, we are going to continue the discussion to show you how we can use or how we can perform the P-Civitan test. And after that, we are going to answer the third question. Okay, so if it belongs to human, then it belongs to who? Okay, so to belongs to who, then we can be answer this question by checking the blood group or by checking the DNA. So before I start to talk about something new, so any questions or problem at this moment? So it seems no question. Okay, so before start, just try to remind you that for the chemistry 7460 forensic analysis, so we have a term paper. So that is one of the assessment in the course. So just check the guideline. Okay, so in the BU Moodle, then you can be see that the deadline, okay, will be next Saturday. Okay, so just try to select one of the topic and then write up a term paper and then to submit, okay, through the Moodle. Okay, so I create a link for the submission already. Okay, so without further ado, then we start to talk about the P-Civitan test. So with the use of the P-Civitan test, what you know is we try to add in a known origin anti-serum. So if it is reacted with our blood sample, so targeting the, let's say the human anti-serum will be targeting the human antigen. If we can see the precipitation, then that will be somehow we know, okay, that is from human. If now, okay, we have an unknown blood sample, we are now adding in an anti-serum that is of a cat's origin. If you can see the precipitation, then you know, okay, the blood will belong to a cat. So therefore, that will be somehow like an try and error process. So actually, at the very early beginning, we only know that this red color stain, this red color liquid is blood, but we don't know what kinds of origin, okay, is it. So therefore, we just try to add in some known origin anti-serum in order to know, okay, so where it comes from. So in order to perform the P-Civitan test, okay, we have three different ways. So we have the capillary method, we have the gel diffusion method, we have the electrophoretic method. For each of the method, it has, okay, some advantages and some disadvantages. So I hope, okay, you can know how we can be comparing these three methods. Then at some situation, then you can select the most appropriate one, okay, to perform the test. So let's start with the capillary method. So although, okay, we say that that is a capillary method, but we are using a, let's say, a test tube, okay, in order to conduct the test. So let's say right now we are using a test tube. So in the test tube at the very early beginning, so we try to add in the anti-serum, okay, so to the test tube. So let's say that is from human, okay, so that is the anti-serum, okay, so from human. So on top of it, okay, we try to add in our blood sample. So we just try to add the two reagent one by one, okay, so one over the other. So if right now we can see some precipitation, okay, that is being seen, okay, at the interface of the two layers. So actually, you know, okay, so the blood, okay, will belongs to human, okay, because the anti-serum from human will only react to human antigen to be resulted with the precipitation. If you can see the participation here, then you know there is a human antigen there, then that will be, the blood will belongs to human. However, if you can't see any precipitation, okay, then you try to change, okay, to another anti-serum to see when you can see the precipitation in order to know, okay, so the origin of your blood sample. So by doing this kinds of capillary method, actually what you can be see is if you are quite lucky that is coming from human, then you know the answer somehow at your first trial. Otherwise, you need to perform the test, okay, for 2 times or 3 times or to 4 times in order to check for the origin. Since, okay, by using the capillary method, you cannot achieve, okay, any simultaneous analysis. So that will be doing one by one. The other thing that you can see is right now we are making use of a test tube. So we first add in the anti-serum, then we try to add in the blood sample. So quite obvious, you need some amount, okay, of the sample, okay, to be dealing with the test. So what you see is the sample size, okay, would not be that small, okay, maybe for some tiny amount of sample, it is quite a lot already. So therefore, for the use of the capillary method, what you can be imagine is if you are dealing with tiny amounts of sample, most probably by the use of the capillary method, that is not that ideal. But never say that that will also always having all kinds of disadvantages. By the use of the capillary method, you can see that it's quite simple, that is quite easy to be performed. So just one test tube and then add the two solutions together, then you can be seeing the result. So therefore, you have some advantages on the easy or somehow very fast, but it consumes lots of sample and also you don't achieve any simultaneous measurement there. So in order to get some improvement, okay, so from the capillary method, then people try to make use of the gel diffusion method to overcome, okay, so two limitations from there. So one that is too much sample. The other is the simultaneous analysis. So by the use of the gel diffusion method for the basic setting, so you are going to make use of a agar gel, okay, so for the diffusion medium. So at one side, you will create a well, okay, and then for another side, you will create another well there. So for the right hand side, you try to fill up with the anti-serum. And then for the left hand side, you try to fill up with the blood sample. So just, okay, when you're seeing the size, actually the sample that you consume will be quite small in an amount. So you can be seeing, so the blood sample, they are concentrated in the well. And then for the anti-serum, they are concentrated in another well. So right now you have a concentration gradient that those kinds of material substance will diffuse out, okay? Because only at the center you have those kinds of substance. At the outside part, you don't. So therefore, they will diffuse out, and then finally, they will be meeting each other. If they are matched, okay, towards each other, then you will see a line of precipitation in between. So therefore, if you can see, okay, a line of precipitation, then by knowing what kinds of anti-serum that you have added, okay, then you know the origin of the blood sample. So therefore, by the basic setting, quite straightforward that you have been solving for the first limitation arising from the capillary method, okay? So right now, small amounts of sample can be used. How about for the simultaneous measurement? So basically, you can change the setting a little bit. So you still are making use of the agar gel, okay, as a diffusion medium. So right now you can make a well, okay, at the center, and then you can make another well, so at the side. So most likely at the center, you are going to fill up with the blood sample. And for the side, okay, so you can be filling, let's say, on top, you are filling the anti-sebum of the human origin. On the left-hand side, you try to fill up the anti-sebum that is of a cat origin. At the bottom, you try to fill up the anti-sebum of a dog origin. Maybe on the right-hand side, you try to fill up the anti-sebum of the rabbit origin. So therefore, you can now, okay, having 4, okay, anti-serum, and then you do a measurement at once. Since, okay, everything are just concentrated in the well. So because of the concentration gradient, they will diffuse outward. So the blood, okay, will diffuse outward and then getting to the well of the anti-serum. And then the anti-sebum, and then they will also diffuse out, try to getting in touch with the blood sample. So when, okay, they meet each other, if they match, then they will be seeing some line of precipitation in between the two wells. For example, right now, if we can see a line of precipitation that is somehow on the right-hand side, so what kinds of origin, okay, for the blood sample that we have? So give me a reply, okay, in the chat room. Okay, so if now you can see, okay, the line of precipitation that is shown on the, let's say, at the right-hand side. So what kinds of origin, okay, do we have? Not a cat, okay, the cat that is somehow located at the left-hand side. So right now we are seeing the line of precipitation on the right-hand side. So it should be a rabbit. Okay, so the rabbit anti-sebum that is put at the right-hand side. So if they meet each other, then they match, then you can see the line of precipitation. Okay, so therefore right now what you can be imagine is you have one sample and you are doing the measurement simultaneously. It will not be a try and error analysis. So therefore one setting, one experiment, then you can be just seeing the position of the line of precipitation, then you can know, okay, so what it is. For example, like if you are now seeing the line of precipitation that is on top, so on top, we are putting out with the human anti-serum, then you know, okay, line of precipitation here, then you can find a human antigen from the blood sample. So obviously, that will be the blood coming from human, okay? So therefore, right now, what you can be see is by the use of the capillary method, that is quite fast, it is quite easy, but it consumes a lot of sample. At the same time, you do not have any simultaneous measurement. By the use of the gel diffusion method, actually you overcome, okay, the two disadvantages, small amount of sample and the simultaneous measurement. However, by the use of the gel diffusion method, the time needed that will be quite long, relatively long, okay? Because right now what we are saying that is a diffusion. If you know about the diffusion coefficient, the diffusion rate, actually it takes some time, okay, for the blood sample and the anti-serum to diffuse out, okay? After they diffuse out and then they need to meet each other before seeing the result. So therefore, for the jet diffusion method, it takes you a long time, okay, so to do so. So in order to speed up the analysis, okay, under the agar gel setting, we try to install 2 electrodes. So one at the left-hand side, most likely that will be negative. And then we put up another electrode, most likely that will be positive on the right-hand side. So we still are drilling 2 wells. Okay, so one at the left-hand side, the other on the right-hand side. So on the right-hand side, we put with the anti-serum. And then for the left-hand side, we put with the blood sample. So right now we have the electrode, we have a potential in order to push, okay, those kind of antigen and also anti-serum migrate towards each other. So once they meet, if they match, you will be seeing the line of precipitation. So right now with the influence of the electric field, with the apply of the potential, actually you speed up the process. So the time needed will be relatively short, okay, when we compare to the original jet diffusion method. But right now you lose, okay, the capability for the simultaneous measurement. Because under the electrode setting, it is hard for you, okay, to drill up with the this configuration. Okay, if you are drilling up with the center well and then the well at the side, actually it is no way for you to put up the electrode and then influence with the electric field. So therefore, right here, what you can be see is for the capillary method, that is easy, that is fast, but it consumes a lot of sample and you don't have any simultaneous measurement. For the gel diffusion method, you have the capability for simultaneous measurement and you consume just a little bit of sample, but that will be quite slow. Under the electrophoretic method, that will be fast and also you consume just small amounts of sample, but you don't have the simultaneous measurement. Therefore, when you are comparing the 3 different ways, okay, to perform the precipitation test, actually you don't have a way that will be absolutely perfect. Then under your situation, then you try to see which one will be the most appropriate, and then you try to put up the blood sample and then add in the anti-serum of the known origin. When you see the line of precipitation and you know about the origin by knowing what kinds of anti-serum that you have been added. So in the precipitant test, actually you can see one special feature there. So we never trying to mix, okay, the anti-serum together with the blood sample. So when you are looking at the capillary method, we try to have the bottom layer for the anti-serum and then the top layer for the blood sample. We never shake it or make a raw test in order to mix it together. And for the gel diffusion method or for the electrophoretic method, we just try to make the blood sample and the anti-serum diffuse, okay, towards each other. And then once they meet, okay, then they are matched, then you can see the precipitation. We never try to mix, okay, the 2 solutions. We just try to let them diffuse and then get in touch. Actually, by knowing the anti-serum, that is certain kinds of antibody, and then we have the antigen there, actually they are all protein. If you are quite familiar with the protein chemistry, actually if one of it, okay, that is somehow in excess towards another, so you will be somehow dissolved towards each other. On the screen, what you can be see is, if right now we have in excess antibody, okay, so that is in excess of the anti-serum towards the antigen. So the antigen will be dissolved in the antibody. So in another extreme, okay, that is on the right-hand side. So if right now you have in excess antigen, okay, that means you have a limited amount of anti-serum. So actually those kind of anti-serum will be dissolved in the antigen. So actually you will be seeing a false negative result. If they are just dissolved with each other, you can't see any precipitation there. So therefore, we just try to let them to be get in touch only, so that the amount, okay, of the antigen and the anti-serum they are getting in touch will be in an equivalence. So if they are at the equivalence, then when they are meeting each other, they just try to bind together, then you will be seeing the precipitate because of the, they are quite insoluble, okay? So they are quite big, okay? So therefore, never try to mix, okay, the anti-serum and the blood sample. Otherwise, you don't know which one that is in excess towards each other. Then you have a false negative result because, okay, they are dissolving towards each other instead of forming the binding, forming the bonding to get to see, okay, the precipitation there. Okay, so right now we move, okay, to the final question. After confirming that the red color stain, okay, the red color liquid, that is blood that is coming from human. Then at last, okay, we try to achieve the criminal identification. That is the personal identification to know, okay, so who it belongs to, okay, so, and then we know about, okay, the blood. So by doing so, actually we have two ways, okay, to do it. The first way that we do that is by the use of the blood group. Because what you know is from the blood, okay, we can be classified into the A, B, AB, and O. So indeed, okay, that is a certain kinds of class characteristic, but we still can get some connection, okay, between the sample that we have, the physical evidence that we have, to be connected with the victim or connected to the murder. So imagine that in the crime scene, okay, we find some blood stain that is of a blood group A. Then you know, okay, so from the case, the victim, okay, his or her blood group that is A, then you know, okay, that kind of blood stain most likely that will be belongs to the victim. So by matching the blood group, okay, then you can be probably connect, okay, the physical evidence towards a person. Or right now, from the cases, from the suspect and from the victim, let's say we find the blood group A, okay, from the red color stain, okay, the blood, okay, at the crime scene, that is blood group A. But the victim and the suspect, none of them, okay, that is the blood group A. Then you will be quite curious, okay, so who have been left behind this blood stain, okay, with the blood group A, then you can know there is one more person being involved, okay, so you know, okay, you miss someone, okay, because the blood that you have at the crime scene, that is blood group A, and then the victim and also the suspect, none of them, they are blood group A, then you know, okay, you have been missing for a person, okay, then you can be having some further information to deal with the investigation. So to deal with the blood group, most probably we are making use of the agglutination test. So for the agglutination test, from the name you know, for a positive result, you are going to see with the agglutination. So for the observation of the agglutination, I will show you somehow after by talking about the respective principle. So for the agglutination test, most likely we are involving with the antigen. And then we can be making use of some antibody. So if they are matching with each other, then you will be seeing the agglutination, okay, will be there. For the agglutination, most likely what you can be imagine is if the antigen and the antibody, they match with each other, so they will become, okay, together. So that's why, okay, when you are seeing, if you have the red blood cell, okay, the red blood cell on the surface, you can find the surface antigen, okay. If you have the surface antigen A, then that will be blood group A. If you have the surface antigen B, then that will be blood group B. So if you have the antigen A and B together, then that will be the blood group AB. If you don't have the surface antigen, then that will be blood group O. So just imagine that you have some surface antigen from the red blood cell. So if you are adding in some antibody, if they are matching to the antigen, so let's say you add in the antigen A, so into the blood sample, okay, if the blood sample, they are blood group A, so it contains the antigen A and antibody A, they meet with each other, then they will be just combined. Okay, if you have the antigen B, then you have the antibody B. If they match, okay, then they will combine. So once, okay, they could be combined towards each other, then you will be seeing those kinds of red blood cell, okay, they will be just come together, just come to aggregation, okay, they will be aggregate, okay, towards each other. So to forming some red color spot, okay, in the sample, because those kinds of red blood cell that come together, then you will be seeing a very intense red color. So next is the pressure, okay, being aggregate towards each other, that is quite high enough. So finally, those kind of red blood cell will burst so that the red color will be disappear. So therefore, the agglutination in terms of the observation, you will be seeing two. You will be seeing the red color spot, okay, has been formed, and then the red color spot, okay, will be disappear finally. So just try to let you see, okay, the positive result, okay, of the agglutination. So right here, what you can be see is for the negative result, the blood sample will be still intact. Okay, so you will be seeing the red color like white as a whole. So if right now it is a positive result, okay, by adding in the antibody, if it is being matched with the antigen, then the red blood cell will be just come together, being aggregate. Then you will be seeing individual red color spots. And then at the end, those kinds of red color spot, okay, will be disappear. So therefore, that is a positive result for the agglutination. But right here, what you can be imagine is the test, okay, will be running individually because you cannot have a mix of the antibody, okay, in order to chat up with the blood group. So antigen A will be matching to antibody A, and then antigen B will be matching to the antibody B. So therefore, you have to add in the antibody somehow individually by the combine of the result in order to know what kind of blood group, okay, is it. So let's say for the antigen, okay, we assume it is coming from your sample. So you have the blood sample. So you are checking for the surface antigen of the red blood cell. So what you try to do is we try to add in the antibody A, okay, to the sample. So next, okay, we try to add in the antibody B to the sample. So we also try to add in the antibody D, okay, to the sample. Because for the antibody D, they will be telling us whether the blood group that is positive or negative. Because we have the A positive, A negative, so that kinds of antibody D will further classify the blood, okay, into 8 different classes, okay? So right now, what you can be imagine is if we are adding in the antibody A, okay, to the sample. So if it is showing you with the positive result, so meaning that in the sample, it contains surface antigen A. So if we are adding in the antibody B to the sample, if it is still positive, then you know, okay, in the sample, it contains surface antigen B. So if we are adding in the antibody D to the sample, so if it is also positive, that means, okay, we have the antigen D, okay, as the surface antigen in the sample. So right now, in the sample, it contains antigen A, antigen B, and the antigen D. So that we can be saying the result is the blood group that will be AB positive. So just read the result directly. It contains A, it contains B, it contains positive, and the AB positive. So in another way, if right now we add in antibody A, that is a negative result. If we add in antibody B, that is positive. If we add in antibody D, that is a negative result. Then the blood group that we have will be the B negative. Because from the result of the antibody A, so we don't have any antigen A there. Then from the result of the antibody B, then we know we have the antigen B there. So therefore, that will be blood group B. And then for the antigen D, which is negative, so that will be a negative blood group. So right now, that is the time for your practice. So I know that will be a very easy task for you. So if now we have the negative for antibody A, negative for the antibody B, positive for the antibody D. So what kinds of blood group, okay, does the result show? So negative for A, negative for B, and positive for D. Okay, so what kinds of blood group, okay, is the result showing? Okay, so everyone get a correct answer. So that will be blood group O positive. Okay, so because you don't have A, you don't have B, okay, then that will be O. And then you have D there, then that will be positive. So how about if right now we have a positive for antibody A, negative for the antibody B, and then negative for the antibody D. So what kind of blood group, okay, does the result show? So positive for A, and then negative for B, and then negative for D. Okay, so you know the answer is quite straightforward. So that will be A negative. So therefore, what you can be imagine is for the test, okay, it won't be just perform it once, okay, because you have to add in the A, B, and D, okay, so individually. So therefore, at least you have to perform the test, okay, for 3 times. So for the first time, there will be A, second time, there will be B, and then third time, there will be D. At the same time, you need a control, okay? Otherwise, okay, you don't know whether the agglutination that is somehow by default, okay, it happened. So you need the fourth one as a control. If you're seeing a positive result at the control that is positive, then the result will be invalid. So for the control, it should be always negative in order to confirm, okay, or validate the result that you have for the antibody A, antibody B, and the antibody D. So therefore, after you know the blood group of the blood sample, so basically you can make a very simple connection. So whether that is belonging to the victim, belonging to the murderer tentatively, or you are now having some missing person, okay, so from the crime scene. So when you are knowing with the agglutination test principle, so actually we have the antigen antibody binder that will be agglutination. So if we are making use of the blood sample to checking for the surface antigen, then we try to add in the known antibody for the reaction. However, by checking with the blood group, okay, so last time I have already told you that to check with the blood group, it is not only by checking with the surface antigen from the red cell. Actually, we can checking the antibody in the plasma. So that will be the liquid fraction of the blood sample. We check for the respective antibody. So how about now we just try to make an other checking. For example, now we say that, okay, the antibody that is coming from our sample, okay, we check for the antibody, okay, in the plasma. So how we can be able to add in the antigen, okay, into the sample. So to doing so, we try to add in something that is the known red blood cell. Okay, so we can buy some known red blood cell in order to add into the sample. For example, we try to add in the known blood cell A, okay? And then we can also buy another kinds of known blood cell. So the blood cell, let's say that will be B. So that is the 2 kinds of reagent, okay, that with a known antigen there. With the known blood cell A, then you know you are adding in the surface antigen A, okay, towards the plasma. With the use of the known blood cell B, then you are knowing you are adding the surface antigen B, okay, to the plasma. So actually, if they are matching with each other, you will still be seeing the agglutination because the plasma, okay, it contains certain kinds of antibody. Then they will be bind to the blood cell will still be aggregate, and you will be seeing the red color spot, and then at this last, okay, they will be disappear because they burst finally. So therefore, there will be another way, okay, to pay with the agglutination test. So how about right now, if we have the positive result, okay, so from the known cell A, and then we have the positive result from the known cell B. Okay, so for the blood group, that will be of the blood type O. Okay, basically, if now we have the known cell A, okay, of a positive result, so that the antibody that we have in the plasma, that will be antibody A, and then with the positive result from the known cell B, you know in the plasma you contain, okay, the antibody B there. So in the plasma, you have antibody A and antibody B there. So in a reverse, that is on the surface antigen of the unknown red blood cell that you have, it contains nothing. So therefore, it contains nothing, that will be blood type O. So therefore, it will be kind of a reverse logic, okay, in order to tell, okay, about the blood group. For example, like if now we have the negative result from the known cell A, and then with a positive result from the known cell B. So actually the blood group that we have will be of the blood type A there. Because you don't have any antibody A there, but you have the antibody B in the plasma. So in the plasma, you have the antibody B, meaning that for the red blood cell, that will be have the surface antigen for A. That will be in an opposite manner, so that the blood group that you have will be of the blood type A. So how about if we are now having the result for both negative? So negative result for the known cell A, negative result for the known cell B. So what kinds of blood group, okay, does it show? Okay, so thanks for your reply. So the result, okay, will be showing you with an AB blood group, okay. That will be quite straightforward, okay. So you don't have any antibody A, then you don't have any antibody B, then actually you don't have these two antibody. In a return, you have these two, okay, on the surface antigen. So antigen A and B there, then that will be blood type AB. So for the final example, positive for known cell A, negative for known cell B. So that will be quite straightforward. Okay, I know you know the answer somehow immediately. So that will be blood type B. Okay, because in the blood type B, you have the antibody A. Okay, so in the plasma. Okay, then in the reverse, then you have the surface antigen B. Okay, on the red blood cell. So therefore, you can be paying with the agglutination test in two different ways. So either you add in the known antibody to the blood sample, or you try to add in the known blood cell to the blood sample. By observing the result that you have, okay, still there will be a combination of results. Then you can be knowing what kinds of blood group, okay, so that is showing. So actually here, that will be get to a comparison. So which way would be better? By adding in antibody or by adding in the blood cell? Okay, so which one that is better than the other? Or what I say is which one that will be preferred over the other? So adding in known antibody that will be preferred, or we add in a known blood cell that will be preferred. So any idea, okay, from student? Okay, so you know the answer. So by adding in the known antibody, that will be better than adding in the known blood cell. So actually by seeing the result, okay, you know. By reading the result, okay, of the known antibody, that will be quite straightforward. You read it directly. So you have A, you have B, you have D, then there will be AB positive. You have B only, okay, then there will be B negative. Actually, that will be a quite straightforward reading of the result. But by using the blood cell, that will be in an opposite. So positive, positive, that will be nothing. Negative, negative, that will be both of it. Okay, so therefore the result, okay, in terms of the reading is direct reading. And then that will be indirect reading. So the other thing that you can see is by using the known antibody, you can be classifying the blood into 8 different types. You have A positive, A negative, B positive, B negative, AB positive, AB negative, O positive, and O negative. But by using the blood cell, so you can be only classified down to A, B, and O. So actually, in terms of the discrimination power, by adding in a known antibody, that will be better. So the other thing that will be quite minor, okay, by using known antibody, that will be cheaper than using the known blood cell. So therefore, altogether, most likely, by using the antibody, that will be preferred over the known blood cell. Okay, so that is how, okay, we can be answering the third question, okay, by the use of the blood group. Just applying the agglutination test, and then you can be tell what kind of blood group it is showing. So actually, apart from the blood, okay, blood group can also be achieved by the other kinds of body fluid. So what I mean is, not only blood, okay, can be go forward with the 3 questions, then we can know about the blood group. Actually, by the use of the other kinds of body fluids, you still, okay, can be going down to the last question, and then you can identify someone by the use of the blood group. Because in the world, okay, there are 80% of the population, we are saying that we are all secretor. So secretor meaning that the blood group antigen, they are also present in the non-blood body fluid. So those kinds of blood group antigen, they could be still be appear, let's say in your tear, in your sweat, in your urine, in your saliva, in your stomach juice, in the semen, or somehow all kinds of non-blood body fluids, you will still finding those kinds of blood group antigen. So therefore, if you are now getting different kinds of body fluids, if you still, okay, want to be seeing, so who, okay, it belongs to, so you still need to go through the 3 different kinds of steps. So let's say if you are now using saliva, then you need to ask whether that is saliva. So if it is saliva, then it belongs to human. So if it's a human saliva, then we try to know, okay, so who it belongs to. If now you find the semen, okay, then you try to first ask whether that is semen. So if it is semen, then it belongs to human. So if it is human semen, and then they will be seeing the blood group and then to know, okay, who it belongs to. So actually the step, they are exactly the same, but the test, they will be a little bit different, especially to answer the first question. For the second one, we still are making use of the P-Zivitin test to checking for the human antigen. And then for the blood group analysis, we are still seeing those kind of antigen there, so they are the same. But for the first question, is that body fluid exactly the same as that body fluid? Then we have to seeing the differentiable characteristic. If we are talking about blood, okay, for the blood sample, we are checking with the hemoglobin. So hemoglobin, that is the unique characteristic for the blood. So how about the other? For example, like if you are now talking about saliva, so what kinds of differentiable characteristics are there? So most likely in the saliva, you will be seeing the amylase. So amylase, that is the enzyme to digest starch. For the other kinds of body fluids or the other kinds of colorless liquid, so obviously it won't, okay, contain any kinds of amylase. If you can be checked that that kinds of liquid contain amylase, most likely there will be a saliva. So in order to check with the saliva, we try to see whether it contains amylase there. So in order to check with the amylase, most probably we try to add in starch. If you add in starch, okay, the starch could be digested. So most probably that fluid contains amylase there. So therefore, we have the 2 different kinds of tests, okay, to test for the amylase by using starch, okay, so there. So one is the Barbitis test, the other that is the starch iodine diffusion test, okay. But for the details, okay, I'm not trying to bother with you, okay, but just try to tell you the differentiable characteristic that the 2 tests, they are targeting the amylase, okay, by adding in starch. So with the starch, with amylase being digested, then probably you will be seeing the positive result of certain kinds of color change. So for the semen, okay, that is another kinds of body fluid that we can find, okay, at the crime scene, especially in some sexual cases. So for the semen, we are checking with the sperm cell. So for the sperm cell, that will be the unique characteristic, okay, for the semen. Because for the other kinds of body fluid, okay, you can't be able to find any sperm cell there. So for the sperm cell, it contains a high amount of protein so that you can be checking by the UV lighting. So you will be given with the fluorescence. Or even you just look under microscope, okay, because you know for the sperm, okay, you have the head and the tail. Actually, the shape, okay, the appearance is quite unique. So by identifying the sperm cell, then you know, okay, that is a semen, okay. So therefore, after by analyzing amylase for saliva, sperm for semen, or even hemoglobin for blood, then you try to check for the precipitin test for the human origin, and then check for the blood group with the agglutination test. So therefore, you can be know, okay, so who it belongs to. But at this stage, okay, you can still be quite curious or quite strong enough to have a strong opinion to say. So by analyzing blood group, that will still not be quite perfect because it is belongs to a class characteristic. So maybe now, okay, for the victim, for the murder, both of them, they are belonging to the blood group A. Then the blood stain that you find at the crime scene that is also blood group A. Then the blood stain belongs to the murder or belongs to the victim. Actually, we cannot tell. Because for the blood group that is a class characteristic, pinpoint to a population only. So therefore, in order to make the analysis to be more unique, okay, so we try to answering the third question by analyzing the DNA. So no matter you are using blood, okay, you can be analyzing DNA by the red blood cell. And then for the saliva, for the semen, actually you will still be finding the DNA information. So therefore, to be more unique, okay, to be much more 100% guaranteed that that is that person, DNA analysis would be performed. So right at the time, okay, so I will let you have a 10 minutes break before we further talk about the DNA analysis. Okay, so let's return from the break. Okay, so can you still hear from me? Okay, so I assume everything's still working. So just answer one question during the break. Okay, so for the non-cell, one student asked, okay, whether we can add in some non-blood cell in order to determine any antibody D that is present in the plasma. But actually, if you want to make it quite clear, okay, for the result that is really the antibody D, then you have to add in a non-cell D, okay, so from the red blood cell. Okay, so but you know, okay, so from the red blood cell, okay, we don't have a red blood cell that is of a type D. So therefore, we don't have such kind of approach, okay, to check for the antibody D, okay, in the plasma. So that is the reason why we can only add in the non-cell A and non-cell B there in order to check for the antibody A and the body B in the plasma. Okay, so I hope I have answered your question. And then right now, okay, we are going to talk about the DNA analysis. So before I go forward, so let me clean up the whiteboard on the left-hand side. So for the DNA analysis, or we say we try to analyze the DNA profile, okay, so of the sample. So that will be an individual characteristics, okay, so that if you are knowing that the DNA that is the same, then quite obvious that will be belongs to that person. So therefore, the result that will be unique, and that is an individual characteristic, and quite straightforward to draw the conclusive conclusion. At the same time, for analyzing DNA, we have a lot of advantages. So such as the DNA that is quite robust, okay, then it will still could be surviving under a wide range of extreme environmental conditions. So what it means is in the blood, okay, you have the DNA actually coming from the white blood cell, and the blood group that is from the red blood cell or from the plasma. Actually, for those kind of surface antigen or for those kind of antibody, they could not be survived for a long time. For example, you can be imagined that if the blood that is dry, so you don't have any plasma anymore, so you cannot, okay, be retrieving with those kinds of antibody in the plasma, or at the same time, those kinds of red blood cell, okay, that has been burst, okay, then you don't have any surface antigen to check for the blood group. But for the DNA, so no matter the white blood cell that is dead or not or burst, actually the DNA they will still be remain intact. Okay, so there. So therefore, there will be somehow DNA can be preserved for a long time that you still can be able to check. Okay, for the respective identity. So for the DNA, no matter you are checking for different body fluid or from different kinds of biological sample, let's say in different kinds of organ. So actually the DNA will be the same. Okay, so therefore there will not be any kind of deviation that since okay there will be an individual characteristics. So once it match, okay, then you match okay for the person already. So nowadays the technique for the DNA analysis that is quite good enough. Okay, you know we have the PCR, we have the electrophoresis. Actually, they are being used for the DNA profiling. So at last, okay, for the DNA result, it could be compared with something that appear in the database. What it means is, if right now you don't have any reference sample from the victim or from the suspect, actually the result being analyzed could be compared with the database. Actually, you still can be matching for the identity. So actually, that is one of the hurdle, okay, let's say in the comparison. You need, okay, some reference sample, okay, for the comparison purpose. For the DNA, if you have the reference sample, then that will be quite perfect. But if you don't have the reference sample, you still can be checked, okay, with the database. You still can be achieving your goal. For the DNA, it could be appear in different kinds of biological sample. So you can see from the blood, it will be appear in the red blood cell, in the semen, that will be in the sperm cell, in the hair, that will be in the follicle, on the skin, that is the skin cell. So on the sweat, you have the skin cell as well. And for the urine or etc., you have the muscle cell there. So once, okay, you have the living cell, then you will have the DNA there. But the DNA on the screen, actually you can see we have two types. So we have the nucleus DNA and we have the mitochondria DNA. But you know, okay, nucleus and the mitochondria, they appear in the human cell, in the living cell. So no matter what, okay, you have the cell, okay, then you can be finding the DNA there. But what we can be say is, from the nucleus DNA and from the mitochondria DNA, the differentiation power that would be different. Because from the nucleus DNA, that is really the one that coming from your father and mother. So that will be a unique combination, okay? So your nucleus DNA, that will be a unique combination from your father and mother that is really unique that in the world unless you have a identical genes. Otherwise, the DNA would be different from the others. For the mitochondria DNA, that is only coming from your mother. What it means is you will have the same mitochondria DNA with your mother. So at the same time, if you have brother or sister, actually you will share the same kinds of mitochondria DNA. So if you try to extend a little bit, okay, on the family scope, actually your mother's mother, that is your grandma will also have the same kinds of mitochondria DNA with you. So maybe your mother has her brother or sister, actually the mitochondria DNA will still be the same. So therefore, if we are talking about the mitochondria DNA, that will be getting to the family of your mother. So meaning that if you are checking for the DNA, nucleus DNA, it is really, really unique. That is really the individual characteristic. But for the mitochondria DNA, that will be, let's say, like a class characteristic, just pinpoint, okay, to a group of population that will be the family of your mother. So therefore, if it is possible, by checking the DNA and the DNA that we are analyzing, that is the nucleus DNA instead of the mitochondria DNA. So for the DNA, what you know maybe from the high school biology, you know it is a polymer. The monomer, that is a certain kinds of a nucleotide. For the nucleotide, it will contain a sugar ring there. And then at one side, you will have a phosphate group. And then on the side group, you have a nitrogen containing group. Then just depending on the side group, that is the nitrogen containing group, then you know what kinds of nucleotide it is, okay? So basically, we have 4 different kinds of nucleotide in human. So to be a simple word, we have the A, we have the G, we have the T, and we have the C. So therefore, with the backbone from the sugar molecule together with the phosphate group, then we try to read along, okay, the side group in order to tell, okay, the DNA sequence. So as you know, for the DNA, that is a double helix structure. So you have opposite strings there. And the A and T will be a match, and the C and G will be another match, okay, because of the match of the number of hydrogen bonds. So therefore, for the DNA, basically, you can just, okay, read it, okay, from the side group. Maybe from person, let's say from person number 1, you will, okay, have the sequence, let's say for the A, T, C, C, T, G, C, T, A. Maybe that is the sequence from person 1. And then maybe right now we have another person, okay? Maybe the person will be of the A, T, C, C, T, G, G, T, A. So maybe that is another sequence, okay, from another person. So basically, everyone is different, okay? But we say that is different, that is only contributing to 0.1% difference only. But what you can be imagine is, if you are not checking for the nucleus DNA, everyone is different, and then you can be able to read along the DNA sequence, most probably you can be able to spot out, okay, the nucleotide difference. So at this position, person 1 that is of a C, and then for person 2 that is of a G. So you can be tell apart, okay, who is who. But by checking the DNA by using this kind of single nucleotide difference, it may, okay, be giving you certain kinds of problem, or the analysis will be quite suffer. Because by checking the whole sequence, actually that is quite time consuming. Okay, it will be quite time consuming and also spend a lot of effort in order to review the whole sequence. At the same time, the DNA length, it is quite long, okay? Under the PCR, under high temperature, you may be resulted with certain kinds of fragmentation. Actually, the amplification power would be quite low. If you don't have sufficient amplification under the PCR, in a return, the sensitivity of the analysis will also be quite low as well. Then actually you won't be able to analyze the DNA sequence accurately. So that is somehow what you can do, okay, analyzing the whole sequence to spot out a single nucleotide difference. But the whole analysis, okay, may not be that of high quality. How about we try to make use of another approach, okay, for the analysis. So let's say, could we making use of some enzyme, okay, to cut off, okay, the DNA sequence with some unique, okay, fragment. So let's say we are using an enzyme that will be cutting at the CT region. Okay, so cutting at the CT bonding. Okay, so there's a restriction enzyme. So when you see the CT, okay, then you will cut it once, and then you are seeing the CT cut it at the second time. And then for the second person, you will be seeing the CT at this position. So therefore, with this kinds of restriction enzyme, okay, restriction fragment, so from the person one, you will be seeing we have 3 fragments being created. And then for person two, okay, so you will be seeing 2 fragments that is being created. So therefore, the number of fragments and the fragment length, okay, of each of the fragments, that will be quite different from each other. Because different persons, they have a unique sequence. So under this kinds of unique sequence, the fragment will be different. Then basically, by knowing the number of fragments and the fragment length, okay, of each of the fragments, then we can be identify someone, okay? So it seems, okay, to be quite good enough, okay? So right now, what we have been asking is, originally, that will be quite time consuming to read the whole sequence. The whole sequence is too long to be amplified sufficiently using PCR now, okay? So it's being cut, okay? And then that will be quite good. Being cut will be being shorter. So being shorter, the amplification will be improved. But actually, you may, okay, still are suffering from this kind of amplification problem. Because right now, after you cut, okay, the DNA sequence, maybe some of the fragments, they are still quite long enough. So meaning that if it is quite long, then the amplification power would be quite low. Then you will be missing some of the fragment. So let's say when we are looking at the person two, so the second fragment that will of somehow 1, 2, 3, 4, 5, okay, 5 nucleotide there. Maybe it is even longer. We have something at the behind. So maybe the length it is too long to be amplified. So ideally, we have fragment 1 and fragment 2 after cutting at the CT region. But maybe one of the fragments that is quite long enough, okay, poorly amplified by the PCR. From the result, it will only showing you 1 fragment there. The other that is missing. So therefore now, okay, the result showing you with 1 fragment, ideally that will be 2. Then that will be having some false negative result because now you can't see something that you want to see. So therefore the result that will still be doubtful. Okay, so we cannot be guaranteed that the result that we see, okay, after the cutting, that is really true. Because maybe some of the fragments, they are still be quite long enough that could not be able to amplify using PCR. So therefore, if now we cannot read the whole sequence, we cannot cut the DNA sequence uniquely, then how we can analyze the DNA? Then actually, when we are seeing with the DNA sequence, you know, okay, the DNA that is used to produce certain kinds of important protein in our body. So for some of the region, okay, so that region will be used to produce protein number 1, and then for another region that will be used to produce protein number 2. So for these 2 important sequence, okay, protein 1 and protein 2, actually you will be finding some spacer in between. So this kind of spacer will be used to separate the two important regions. Otherwise, okay, these two important regions, if they are somehow connected directly with each other, maybe our body will miss sensing that, okay, so this is a whole sequence to generate one kind of protein instead of generate two. Then that will be in a disaster. So therefore, our body that is quite clever, okay, we try to separate the important protein region by using those kind of spacer. So those kind of spacer, we say that that is a short tandem repeats. So let's say for person number one, maybe you have a very important sequence number one for this part. And then you will be having those kind of spacer that is the short tandem repeat. Maybe that is repeated as the A, G, C, T, and then A, G, C, T. And then that will be going to the sequence number two for another protein. So for the AGCT and the AGCT, this is repeating pattern. Actually, from our body, that is meaningless. Okay, but it's just try to help to separate the two important regions for protein 1 and protein 2. So maybe now for person 2, so sequence number 1 and then AGCT, AGCT, and then AGCT, and then sequence 2 here. So actually, you can be seeing that different person, they will have different number of repeat. For the person 1, you will see the number of repeat equal to 2. And then for the person 2, the number of repeat that is equal to 3. So actually, you can be making use of the number of repeat in order to differentiate our two persons. So this kind of short tandem repeat actually, by the name that is short tandem repeat, that is short enough, so that it won't be having any kinds of problem for the amplification during PCR. So therefore, you don't have the problem, okay, with the amplification. You will be seeing all the results that you want to see. At the same time, that will be quite unique, okay? So maybe person 1 repeated by 2 times, and then person 2 repeated by 3 times. Maybe we have the third person repeated by, let's say, 6 times. Actually, different person, they will have different repeating. Then you can be knowing that sample that is belonging to who by the number of repeat. Actually, it is quite funny that, okay, we are analyzing some meaningless region in order to obtain the meaningful result. Actually, to our body, the short tandem repeat region that is a spacer, actually, we don't use it. It is just, okay, a separation between the two important sequences, but we are analyzing this region in order to identify someone, okay? So therefore, for the DNA analysis, so what we try to say is that is the strDNA analysis. So in order to analyze the nucleus DNA, so we try to perform the short tandem repeat DNA analysis. So mainly to saying to you that we are analyzing the short tandem repeat region. Instead of we are reading the whole sequence, or we try to make use of the restriction enzyme. So we just try to look for the number of repeat. So for the strDNA analysis, it will go through 6 different steps. The first step, that is the collection of sample and also certain kinds of preparation. So secondly, we try to deal with the DNA extraction by the use of the solid phase extraction. So after we extract out the DNA from the sample, so we just try to deal with a preliminary quantification. So by making use of the fluorometry. So in order to know about how much DNA copy are there, because if you have a lot of DNA, then for the amplification using PCR, and then you can be just using a few cycles. If you have very, very tiny amount of DNA there, then you need, okay, a lot of cycle to amplify a lot. So therefore, this preliminary amplification quantification that is needed. So after the quantification, then we try to go through the amplification by making use of the PCR in order to amplify the amount of DNA that we have, okay, so that that will be sensitive enough, okay, for the analysis using the electrophoresis. Okay, so the analysis will be done by the electrophoresis. So at last, okay, with the pattern being generated from the electrophoresis, so we just try to look at the pattern, and then we can make use of the comparison directly. So no need to do further interpretation or whatever. Once, okay, you have the result coming from the electrophoresis, so just look at the pattern, and then you can compare side by side. Then you will know, okay, this sample or this kinds of body fluid that is belongs to who. So therefore, we have to go through the 6th step. And then for the first step, basically try to avoid contamination, because you know, okay, everybody from your body, okay, so you will bear with the DNA information. If you are touching the sample with your bare hand, you have the sweat or you have the skin cell, actually you're contaminating the sample. And also avoid degradation, okay, because it will still be degraded, okay, the DNA still can be dyed, but it can be survived for a long time, but it doesn't mean it won't die. So therefore, try to put it into a refrigerator in order to lower the temperature. For the physical evidence that we need, okay, most probably, if you are seeing the body fluid, then that will be quite straightforward. However, for some kinds of object as the physical evidence, you still can be used in order to retrieve, okay, the body fluid for the DNA analysis. For example, like if you are now having the cup or the bottle, actually at the mouthpiece, okay, so when you drink the water, so you will let behind your saliva. So therefore, you need not to be seeing the saliva directly, but if you are sampling at the mouthpieces, actually you can be grabbing the saliva, then you can know about the DNA. Or for example, like nowadays, everyone that is wearing a mask, okay, so wearing a mask. So in the mask, okay, in the side that you are wearing the mask, you will have the breath or you have the saliva. So actually, by checking the mask, you still can find the DNA. Or for someone that is wearing a hat, okay, so you have the cap or the hat, okay, so the hat or the cap. So actually, the hat or the cap may be containing hair or some sweat there, or even from your glasses, okay, so on the lens, okay, maybe you have the tear. So therefore, right here, what I try to convince you is it need not to be seeing those kind of body fluids, may not be just seeing the red color liquid or seeing the semen or seeing the saliva. Actually, you can sample different kinds of objects as physical evidence. You still have the chance, okay, to retrieve out those kinds of body fluid for the DNA analysis. So for the DNA extraction, we are making use of the solid phase extraction. So here showing you the step, okay, for the DNA extraction. We first go through the cell lines. What it means is DNA that is trapped, okay, in the living cell. So right now you have those kind of rubber cell, you have the ribose cell, you those kinds of semen. So you need to physically break it in order to make the cell burst, so that the DNA will be just getting out. So that is the first step, okay, for the cell lysing. So after that, we are going to make use of a cartridge that is binded, okay, to the DNA. So therefore, we just make use of an Eppendorf, and then we put in, okay, the cartridge, and then we put the Eppendorf into a centrifuge, making use of a centrifugation force, okay, in order to press down the liquid, so that the DNA copies will be retained on the cartridge. And for the other kinds of material, so they will be just going away. Because when you are physically breaking down the cell, you will have the cell fragment, or inside the cell, you have the protein, you have the lipids. Actually, we don't want those kinds of matrix. So therefore, we just try to retain the DNA, and then we try to reverse the, just try to up and down, okay, so upside down, okay, the cartridge, and then we try to add in the solvent in order to elute out the DNA, so that you have the DNA solution finally after the solid phase extraction. So with the DNA solution, Then we just try to send into the measuring the fluorescence in order to know about the preliminary concentration. So as I mentioned, if you are seeing a lot of copies, then the PCR cycle could be just a few. If you are seeing tiny amounts there, then you need some more cycle in order to amplify the DNA to have a sufficient amount for the sensitive analysis. So for the PCR, right now what we try to amplify, it is not the whole sequence. Pay attention to that. We are analyzing the short tandem repeat region. So therefore, the part that we are interested, that is the STR region only. We are not going to amplify the whole region. We just try to amplifying the short tandem repeat region only. So therefore, in the PCR, we try to add in some reagent to go through the PCR. So most probably, we need the DNA sample. Then we need a primer. The primer, that is the tape, in order to take the starting point, of the amplification. So maybe what you can be imagine, so on the sequence that we have showing on the whiteboard, maybe the primer will be taking at the end of the sequence number 1, then taking the region at the AGCT repeating. So just tag, so where we try to start. Then we have the nucleotide as the raw material for the amplification. But right now, we are not making use of the individual nucleotide. Because for the normal PCR, we are adding in the individual A, individual T, individual G, and the individual C. But in order to just amplifying the short tandem repeat region, we are amplifying with the raw material for that short tandem repeat only. What it means is we are adding in the raw material as the TCGA as a whole. So that is the raw material that we add, so for the extension. So we add one block and then we add another block, so for the extension. So that will be ended. So for the bottom one, so we add 3 blocks and then that will be extended for once already. So for the M side, that is the one that we have with the extension, but no matter what, is we have the primer to take the starting point, and then we have the block, of the raw material to tag on that for the extension. And then 1 will become 2, 2 will become 4, 4 will become 8, 8 will become 16, 16 will become 32. So within several cycles, actually it will be amplified a lot. For the PCR, just try to give you a little bit information that it will be run, in a temperature program. At the start, we try to heat it up at a high temperature so that the DNA string, the double helix will be just falling apart. And then we try to lower down the temperature so that the primer can be tagged on the starting point. And then we increase back the temperature a little bit so that the raw material as a block will be go through the extension so that the DNA sequence will be finally amplified. So right here, what I just try to remind you is the primer will be taking on the starting point, that is the short tandem repeat region. And then the nucleotide that we add, it is not the individual nucleotide. That will be a block that is the one that we want to amplify. So after we have been amplifying the short tandem repeat region, then we can load the amplified solution to the gel electrophoresis for the analysis. So let me clean up some part on the whiteboard and then to show you what happened. So normally we are running with the gel electrophoresis. So we are preparing with the gel. And then on top, so that will be person 1 and then person 2. So on top, we are adding the electrode for the negative side. And then at the bottom, we have the electrode for positive. So that the amplified sequence, the amplified short tandem repeat region will be just run, on the gel. So for the mobility, what you can be imagine is it will be depending on the length. So if right now we have a shorter length, the mobility will be higher. If the length that is longer, then the mobility will be lower. So meaning that they run on top. If the mobility is high, then they will be forming a band somehow close, okay, the bottom of the gel. So let's say for person 1, so you just repeat it for 2 times, okay, so that the mobility is high, then they will be forming a band at the bottom. For person 2, okay, it has been repeated for 3 times, okay, the mobility that is not that high, then the band, okay, will be forming at the upper part relatively. So therefore, you can see, okay, with the band being formed, okay, being shown on the gel electrophoresis, that will be not the same, okay, with the 2 persons. Owing that, okay, the number of repeat that is different, then they will be showing different positions. And actually, you can tell, okay, by the pattern that is person 1 and then that is person 2. But right here, you may ask, okay, does it really be just amplifying one short tandem repeat region and then forming one band, okay, in order to achieve the comparison? Actually, that is not. Because for the comparison, by comparing one short tandem repeat region, it may not be that unique, okay? Because here just showing to you that repeated, twice repeated for 3 times, maybe we can still have the person number 4, okay? For the AGCT repeating, it is still repeated by 2 times. Maybe you are seeing the same kind of band at the same position. Then how, okay, we can be further increased with the discrimination power. Then after sequence 2, maybe you can be foreseeing that you have a sequence 3, okay, for the protein number 3. In between the sequence number 2 and the sequence number 3, maybe we have another kind of repeat. So let's say we have the TTAC, okay, for let's say for 4 times, okay, before it reach, okay, to the sequence number 3. For person 2, it will be having the TTAC. Maybe let's say it has been repeated for 1 time, okay, then we get to the sequence number 3 already. So if we are dealing with the PCR amplification, we also add in the primer, okay, so to test the region, okay, after sequence number 2. We also provide, okay, with the respective nucleotide, let's say for the AATG, okay, so this kind of block, okay, for the extension. Actually, we can simultaneously amplifying different kinds of loci. So let's say for person 1, okay, if we are doing so, repeated for 2 times, okay, for the first ST out, repeated for 4 times for the second ST out, then you'll be seeing one more band, okay, so on top. And for person 2, just repeated by 1 time only, then you will be seeing one band, okay, at the bottom. So maybe we still, okay, 2 band may not be good enough. Maybe we are still seeing another repeating. So let's say after sequence number 3, okay, you are now seeing the GATA, okay, so repeated for 5 times. Then you get to sequence 4. Then for the GATA, now repeated for, let's say, 6 times. Then you get to sequence number 4. So therefore, if we can also simultaneously amplify the STR for the third kind, okay, then you will be seeing one more band, okay, so for person 1, and then another band for person 2. So therefore, that is the way that we are increasing the number of bands, and then the pattern will become much more unique. Actually, by seeing one short tandem repeat may not be that unique, but if we are now have a combination of short tandem repeat, then the discrimination power will be 100%. If right now we have an unknown sample, okay, from the crime scene, so showing with this kind of pattern, so actually you can be able to draw the conclusion. So from the pattern that I draw on this whiteboard, okay, the sample belongs to who? 1 or 2. So quite straightforward, okay, so sample, okay, belongs to person 1 or person 2. Okay, so thanks for your reply. So this is quite straightforward, okay? So one band, and one band. So actually matching to the band, okay, for person 2. Then basically the pattern that you see, okay, after the electrophoresis, just make a direct comparison. Then everything that is done already. So by the use of the gel electrophoresis, it is a quite good enough measurement. But by using the gel electrophoresis, what you can be imagine is you don't have any automation, and you need to prepare the gel, and you need to run the gel, you need to avoid overheating of the gel. Actually, there are lots of precaution and also manpower required. So therefore, can we have a little bit change, still be electrophoresis, but by using another kinds of electrophoresis to run, the DNA short tandem repeat region. So that will be done by the capillary electrophoresis, So for the gel, the separating medium will be the gel, and for the capillary electrophoresis, the separating medium that will be inside the capillary. So I'm not trying to bother with you, for the separation mechanism of the capillary electrophoresis, but what you learn from the separation science, actually it is a kind of chemical instrumentation. So you have the autosampler, then you can run the experiment automatically. So by separating those kind of fragments, so those kind of short tandem repeat using capillary electrophoresis, you still can be imagined that for the shorter length, the mobility will be higher. Then they will be eluted out at the early beginning. So that will be somehow like a chromatogram. So right now, so you're running a chromatogram, so signal versus time. Maybe now that is for person number 1. So for person number 1, so you will seeing 1 band, and then you will seeing 2 band, so at last, So repeated for 2 times, repeated for 4 times, repeated for 5 times. So that is the, let's say the chromatogram for person 1. And if you're running for person 2, so actually you will be seeing 1 peak, at the very early beginning, and then 1 peak later on, and then another peak, so somehow at the end. So therefore, just depending on the length, that will be the mobility issue. So therefore, the chromatogram that you can be resulted, with the capillary electrophoresis, they are not the same with each other. So basically, there will still be the pattern comparison. By changing, from the gel electrophoresis towards capillary electrophoresis, you gain, the power of the automation. At the same time, you reduce your analysis time. Because running the capillary electrophoresis maybe at most that will be 10 or 20 minutes, but running a gel maybe takes you an hour. So therefore, by the change of the capillary electrophoresis, then that will be better. So that is the page, what I have been emphasized is we are not just amplifying one short tandem repeat region, because amplifying one short tandem repeat region may not be that unique. So that's why, we try to amplify for some more. So let's say what you can see on the screen is for the UK system, we try to amplify for 17, short tandem repeat region. And for the US, we try to amplify for 20 short tandem repeat region. So with this kind of number of short tandem repeat region that we amplify, the pattern that we generate. So obviously you will know, from the UK, you will have 17 band there. From the US, you will be seeing 20. Actually, the pattern that you have will be really, really unique. So in the world, you don't have another person that will be generating the same pattern as you. So that the forensic scientist to be say is the pattern from the short tandem repeat analysis that will be 100% pinning point to one single person. So by using the STR-DNA analysis, we have achieving a lot of advantages. Most probably is we are now seeing the nuclei DNA. So basically that will be a unique property, that individual characteristic. At the same time, for the amplification power, that will be good enough that the analysis will be quite sensitive. At the same time, you won't be missing any of the band, because of the amplification power that is good enough. And then we are now simultaneously analyzing different short tandem repeat region. so that the discrimination power that will be perfect enough, okay, to identify someone. At the same time, you can be identifying the sexual identity, okay, by analyzing the short tandem repeat region in the chromosome that is related to the man and the female, okay, so that you can know about the sexual identity as well. At the same time, you can be checking with the family relationship. Because for the DNA analysis in the crime lab or in the forensic lab, it is not only we are checking the physical evidence, those kind of body fluid that belongs to who. Actually, we will also offer the test for the parentage in order to seeing, okay, so the father and mother and the children or the child, are they really, okay, of the same family? Because what we have been saying is, for the nuclear DNA, that is a magical combination from your father and mother. So therefore, from the child, so every band that will be coming from the either from father or from the mother. So therefore, from the children, so the band matching to mother, the band matching to father, the band matching to mother, the band matching to father. Actually, you can be matching the band of a child, okay, towards one side to mother or the other side to father, in order to reveal, okay, so whether they are of a true family. So what happened is, if now you are seeing a band, okay, so coming from the children, okay, so maybe you are now having a band, okay, from the children. So now you cannot match, okay, to the father or to the mother. Then actually that will be somehow in a disaster. Because you know, okay, so this band should be coming from either father or mother, but right now you cannot find any. Then actually, you know, either the father or the mother, that is not the true father or the true mother. So therefore, it will be a way, okay, to offer with the parentage test, okay. So at last, okay, just try to talk about a little bit, okay, for the mitochondria DNA analysis. So somehow by analyzing the mitochondria DNA, you know there will be a class characteristic just pinpoint to your mother family. But why we still want to analyze it, or we still are having the technique to analyze the mitochondria DNA? So basically, in our sample, okay, so for the nucleus DNA and the mitochondria DNA, the amount, they are different. Most likely, we have much more mitochondria DNA than the nucleus DNA. So if right now, the victim has been died, okay, for a long time, actually DNA could be survived for a long time, but it still can be degraded. So for the degradation rates for the nucleus DNA, for the nuclei DNA and the mitochondria DNA, actually they are the same. Imagine that you have 3 copies of the nucleus DNA and 5 copies of the mitochondria DNA. So if it has been died for a short time, actually you can be seeing both of them, and then you can be taking the nuclei DNA for the analysis. However, if the person has been died for a long time, so the DNA will be degraded at the same rate. So the nuclei DNA all degraded, and then for the mitochondria DNA also be degraded, but you still can see 2 copies has been remade. So at the final stage, okay, maybe you will still be seeing the mitochondria DNA. So it is somehow a last tool, okay, that you can perform the analysis. So therefore, you still need to do something. Otherwise, you don't, okay, have any kinds of information there. From the mitochondria DNA that is of a loop that we cannot find any short tandem repeat there. So therefore, we have to go through the whole sequence in order to see the single nucleotide difference, okay, in order to know, okay, so what are the difference, okay, from there. So in terms of the analysis, actually, that is the same, okay. We need to collect and store the physical evidence, extract out the DNA from the solar phase extraction, and then we get a preliminary quantification by the fluorescence. But for the amplification right now, we are not amplifying the STR anymore. Right now, we try to check for the whole sequence, especially at the HV1 and the HV2 region, that is the high per variable region number 1 and 2, where we can be most likely finding the single nucleotide different in order to deal with the comparison. For the PCR, in order to retrieve, okay, with the whole sequence, we try to deal with the Sanger method. After that, we can still be running with the gel electrophoresis or the capillary electrophoresis. But right now, the pattern that we have, we need to go through the pattern in details in order to tell, okay, what kinds of DNA sequence are we showing. So after knowing the correct exact sequence, then we make use of the sequence for the comparison. So therefore, the analysis, it is a little bit tedious, okay, than the short tandem repeat DNA analysis, okay. There will be Sanger method, the pattern, you need to go through the DNA profiling and then to deal with the comparison. At the same time, the mtDNA that is not really that unique, okay. So therefore, if it is possible, go through the nuclei DNA with the short tandem repeat analysis. So everything that will be perfect. So for the Sanger method, I'm not trying to bother with you, okay? So if you are interested, then you can read it by yourself, or you can ask me later on, and it won't be happen in the exam, okay? So no worry about the Sanger method. So after you are amplifying using the Sanger method, run the gel, then you can read the pattern to recreate the sequence. So finally, you can be compared with the sequence so that you can know, okay, so who he is or who she is. Okay, so that is what I want to share with you for the whole course. For the last lecture material, so that is the trace evidence that is for reference only. So that will be a reference for you to write up the term paper. Then the due date, that will be next Saturday. So submit, okay, through the link that I provide to you. And at the end, okay, so just try to provide you a little bit information related to the final exam. So for the chemistry 7460 forensic analysis, I try to make the exam to be of a take-home setting, and that will be open book. What it means is on the date of exam at 7 o'clock, okay, the final exam paper will be appear, okay, in the BU Moodle, in the classroom. Then you download it, and then you try to write up the answer script. And then after 2 hours, so that will be 9, then you try to scan, okay, your answer and then submit. So at the link that I provide to you also in the classroom. So what it means is you don't need to turn on the Zoom or any camera. So I'm not going to chat or monitor any one of you. So just do it by yourself. Within the time limit, then there will be somehow you download the paper, you do it, and then there will be open book. I'm not worried about what you are checking with, okay? So that will be open book. Then finish within the time, submit on time, then that's it, okay, for the final exam. So later on, I will provide you with the guideline for the final exam for the forensic analysis. So also be upload, okay, to the Moodle classroom, and then I will keep you updated. So if you do have any questions or problem during revision, so just ask me. Okay, so wish you good luck, and I believe, okay, for the class, most of you will be graduate soon, and then wish you all the best in the future. Okay, so I will stop talking. So if you have any questions or problems, so just feel free to stay and ask.